Hello, everyone, and welcome to today's webinar titled The Diagnostic and Clinical Impact of SARS CoV-2 Variants. I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. This educational web seminar is sponsored by Biorad Laboratories. Now, I want to invite everyone to participate by submitting your questions during the presentation. To do so, simply type them into the Ask a Question box located on the far left of your screen and click Send. Any questions we do not have time for during the presentation will be captured and responded through the Q&A list that will be posted next week in the Event Resources box, which is located at the Ask a Question box. If you have any technical trouble seeing or hearing this presentation, simply contact us through the Ask a Question box and we will address your issue. As a reminder, this presentation is educational and offers free PACE continuing education credits. Click on the Continuing Education Credits tab located in the top right-hand corner of your presentation window and follow the presentation instructions to obtain those credits. Now let's get started. I now present today's speaker, Dr. Adam Lauring. Dr. Adam Lauring is an associate professor in the Department of Internal Medicine, Division of Infectious Diseases, and the Department of Microbiology and Immunology at the University of Michigan. He received his undergraduate degree from Yale University and his MD and doctorate from the University of Washington in Seattle. Dr. Lauring pursued his postgraduate clinical and research training at the University of California, San Francisco, where he completed medical residency and an infectious disease fellowship and a postdoctoral fellowship in biology. He also served as a chief medical resident in the Department of Medicine, and he has been on the faculty at the University of Michigan since 2012. Dr. Lauring studied the fundamentals of how viruses mutate and how this drives the evolution of poliovirus and influenza virus. His more recent work utilizes advanced sequencing of patient samples to define how viruses transmit in households and escape host immunity. He's using similar approaches to understand how SARS-CoV-2 evolves within the host and spreads between them. Dr. Lauring is a fellow of the Infectious Disease Society of America and a member of the Governing Council of American Society of Virology. His research on respiratory viruses has been funded by grants from the NIH, CDC, and the Burroughs Wellcome Fund, and the Bill and Melinda Gates Foundation. Dr. Lauring, welcome, sir. You may now begin your presentation.
Well, thank you. It's nice to be with you. Today I'm going to tell you about SARS-CoV-2 variants and what they mean in terms of diagnostics, as well as treatment and vaccination. First, my disclosures. And so the objectives for this webinar are to distinguish between SARS-CoV-2 variants of concern and variants of interest, to identify variant attributes that could affect diagnostic testing, and how you can quality control diagnostic testing, and to summarize the latest data on the most notable SARS-CoV-2 variants and vaccine efficacy. So to start off, I think there's naturally a lot of confusion when people talk about SARS-CoV-2 variants. This is just a clip from the New York Times where Eric Topol and others push back and were saying that there's just too much talk about scariants and that there's a lot of discussion about this variant or that variant and what they may mean, but that often leads to a lot of confusion. So my task today is to try to clear some of that up, because there are certain things about the variants that are actually quite important, as I indicated, both for diagnostics and for how SARS-CoV-2 is managed and prevented. So today I'm going to tell you a little bit about terminology and some definitions, go through some of the key variants of concern that many of you have probably heard about in the news, talk about diagnostics, and how variants are detected, either by sequencing or by qPCR methods. And then I'll conclude by talking about how variants affect treatment with monoclonal antibodies, as well as how well the current vaccines work against the most notable variants. So first, some terminology. A mutation is an actual change in the nucleic acid or the amino acid sequence, in this case, of the virus. So we often talk about specific mutations or mutants. Notable ones would be these two that are in the spike protein called N501Y and E484K, which I'll elaborate on in a bit. And then we also hear a lot about variants. And these are, a variant is essentially just two sequences that are different. Importantly, variants can have multiple mutations that distinguish them. For example, the B117 variant, which we've heard a lot about, actually has 17 characteristic mutations. Then there's lineage is often used. And that is essentially a variant and its descendants. So B117 was initially described as a lineage. It was a group of viruses that ultimately arose from one virus, but then it's sort of a family of that virus and its descendants, as in a phylogenetic tree or, by analogy, a family tree. A strain is technically a variant that is phenotypically different. So it's not just different at its sequence level, it actually behaves differently from a biological perspective in terms of whether it causes a different disease or affects our therapies. There was a lot of discussion early on what's a strain and what's a variant. My view is this is basically a garbage term these days because they're essentially used interchangeably. So there's a classification of variants of concern, or VOC, variants of interest, VOI, or variants under investigation, VUI. The WHO and the CDC will classify variants as variants of concern if they are distinct and have reasonable evidence for concerning biological properties. And most notably, these are whether they spread faster, whether they cause more severe disease, whether they complicate therapies or vaccines. And as you can imagine, there's varying levels of evidence here. And so the variants of interest are still important. It's just there's not as how concerning they should be to all of us. This is a screenshot from this website, outbreak.info, which is well curated and updated. I go to it a lot, and it has information on all the variants of concern and interest, as well as their properties and where they are in the world. So this is the biggest, most important variants of concern, at least currently, B117, B1351, B1617.2, and P1, and I'll be going through each of these in order. So as I indicated earlier, there's variants of interest, and then there's also mutations of interest or mutations of concern. So the variants of interest, again, these are not variants of concern, but they're classified as variants of interest, are B1.427.429, which is also known as the California variant, B1.526 and its cousins, also known as the New York variant, B1.617. There's actually several flavors of this variant. I showed you 617.2, which is a variant of concern, and these other ones are variants of interest. And they're generally grouped together as, quote, the India variants. And then there's also P2, which is related to P1, and it's also was first spotted in Brazil. So those are the variants of interest that you'll be hearing about. And then there's various mutations that are important. And many of these mutations are shared across multiple variants. For example, E484K is a mutation that affects how the virus is recognized by antibodies, and that is present on multiple variants of concern. Similarly, L452R and N501Y are on multiple variants. So again, it highlights the issues of what's a mutation, what's a variant, and how they overlap, which will become important when we get to diagnostics. So now I'll tell you a little bit about key variants. The first one is lineage B117, which is also known as the UK variant. In general, we like to move away from naming variants by places because it might not be accurate. And of course, many of these variants are now all over the world. This was the first variant that really caught the world's attention. It was, again, spotted in the United Kingdom over the fall. It arose as what's called a long branch. What you need to know is that it had a lot of mutations when it was first identified. And there has been speculation, and it's reasonable, that this virus could have arisen within an immunocompromised host because it had accumulated so many mutations when it was first spotted. It spreads more rapidly. In fact, the evidence is perhaps strongest for this variant that it actually is perhaps more transmissible or contagious because it spreads through populations quicker than the other viruses, which underlies how it came to dominate the United Kingdom, much of Europe, and of course now the United States. So it's more difficult to contain. It does not appear to vary in terms of age groups that it infects, although there was early speculation along those lines. Newer data suggests that it may cause more severe disease. It's hard to, as you can imagine, it's hard to do these studies. There's lots of caveats and confounders, but the data is pointing in that direction. This is just to show data that came out at the end of December, showing how quickly this virus spread in multiple areas of the United Kingdom. And of course, as I indicated, this has been now recapitulated in Europe and the United States. B1.351 has also been called the South African variant. It also appears to spread rapidly, although the data are not as strong, I think, as for B117, but it's also perhaps less complete because it arose in areas of the world where there hasn't been as much sequencing or as in the United Kingdom. It's in the US and it's currently staying at a low level thus far, and there's been speculation that it and other variants really just can't compete with B117 because B117 spreads so much better than the others that these other variants of concern can't get a foothold in the host population. There's concerns that it's less neutralizable by serum, by antibodies, which I'll get to later in the talk, and that's thought to be attributable to this E484K mutation, which is in the spike protein, which then causes a change in charge and may affect how antibodies bind. P1 is another variant of concern. It's also known as the Brazilian variant. It was identified when there was, in the fall, again, explosive outbreaks in Manaus, a state in Brazil, and also reports of reinfection with this variant. It has also spread fairly quickly, and it also has 3 mutations that it shares with B1.351. These 3 mutations in spike are K417T, E484K, and N501Y. And so these mutations in combination are what make both P1 and B1.351 somewhat concerning. Next, in the last few weeks, we've been hearing a lot about B1.617.2, which is also known as the Indian variant or the double mutant. It has been coincident with a large surge in India. It's not clear whether it's driving the surge or whether it's just the virus that's been around in India. And that's because its relative transmissibility, how fast it spreads, is not really clear. It has 2 mutations that have caught people's interest, L452R, which is in the spike receptor binding domain, and also important for antibody recognition, and P681R, which is in the part of the protein that may have a role in cell entry. And then there's others that related to this that have this E484Q mutation, which is at the same position, but a different mutation, of course, than E484K, which I indicated are in the Brazilian and South African variants. And this is just to show you the quote California variant, 427 to 429, which we've heard a lot about in the US. And these also share this L452R mutation shown at the bottom here in the receptor binding domain. The receptor is shown in purple. And so again, it's indicating that this L452R mutation may affect how the virus recognizes its receptor and how it might be neutralized by antibodies, and also highlights how different variants can share mutations. So how does one identify these variants? Most commonly, they have been identified through sequencing or genomic surveillance. This is just to show the workflow that we use in my lab to identify variants and to perform genomic surveillance in our area. We retrieve samples from our clinical microbiology laboratories. So these are NP swabs that are in transport media. And then we extract the RNA. We then do a reverse transcription to convert it to cDNA and then a multiplex PCR using the ARTIC protocol, which is what most people use, which generates 109 overlapping 400 base pair amplicons spanning the entire 30 kb genome. We then perform a library prep for next generation sequencing. We either use the Oxford nanopore technology platform shown up top, which offers a quicker turnaround for us, about 4 days from sample to data, or we've used the Illumina platform, which has slightly longer turnaround time, but higher throughput. Once we get the data, we do an additional day of analysis. One, we multiplex our samples, so we sequence up to 192 samples at once with barcodes, and then we use the barcodes to demultiplex these to individual sample sequences. And then we use some available online tools, which are fantastic. One is Nextclade, which allows you to identify the mutations in the sequence data and offers some quality control metrics to determine whether your sample quality is good, whether the sequence data is good, etc. We then use Pangolin, which is another tool which you feed in the sequence data and then it gives you the lineage or the variants. So it'll tell you this sequence is B1.1.7, this one's B1.351, etc. It essentially classifies the viruses based on the signature mutations for each of the variants. Now, we've spent a fair bit of time in establishing our sequencing workflows, paying attention to quality control. And this is important because, as I showed on the earlier slide, there's a lot of steps, including reverse transcription and PCR, and then the actual sequencing, where you can get errors introduced and not true mutations, but false positive mutations. This can occur either at the consensus level, so the main sequence of the sample, or when you're looking for rare mutations within a given sample. Like many viruses, SARS-CoV-2, individuals will have additional mutations that arise after they're infected. These will be at low frequency, often at 5 or 10%. And you can sometimes detect these in the sequence data, but again, it's important that attention is paid to quality control. The way we've done this is we've used defined RNA templates and mix them. And so we can start with an RNA that corresponds to the Wuhan strain of SARS-CoV-2, and an RNA that corresponds to a different SARS-CoV-2 sequence. And then we will mix these together so you can get 90%, 10%, or 80-20 mixtures, and then also dilute how much of our input there is to get a sense of sensitivity. So we'll mix those, do RT-PCR and sequence, just as I indicated before, for a real sample. And what we'll do is we'll look in our data for these individual mutations that differ between the two strains. So if we do an 80-20 mix, we'll say, okay, well, we should see these 7 mutations in this lower strain at 80% frequency, and we'll see 20% of the other. And then also we can then see how sensitive and specific our sequencing and analysis is for detecting specific mutations, in mixtures and also at varying inputs. And so what the data has looked like for us is shown here. And the first series of 4 plots, we're looking at sensitivity. So how good are we at detecting mutations at these frequencies at the x-axis, so 0, 5, 10%, 15%. And I indicated there's 7 mutations, so you can see at each frequency how many of those 7 we identified. And then the 4 quadrants represent 4 different inputs of template. So it gets you a sense of if you have a low input sample, so someone who has a high CT value on their qPCR testing, how good are you at finding mutations in that sample? Then we also use these data to look at our false positive rate. So how many times do you identify a mutation that's not actually in there? Because we have defined templates that we use in these studies so that we can see if there's a mutation that we find in our data that's not there, that's a false positive. What we find is we often have a lot of false positives when there's very low input samples. So this one with 100 false positives is a sample that had 100 copies of RNA in it. And so that's not very much, and that the number of false positives reflects that you go through a lot of RT-PCR cycles where errors are introduced when you have lower input sample. And so that is a good way of using defined templates to quality control your sequencing and have confidence in your data so that you can then move forward to actually identify variants. There's also been a proliferation in recent weeks and months of non-sequence diagnostics for variants. Of course, with sequencing, you can find whatever is out there. It's unbiased. But sequencing diagnostics, you can find things that you're specifically looking for, and also with less cost and a more rapid turnaround. This became prominent with B117. B117 has a characteristic deletion in its spike protein gene, and Thermo Fisher's TAC-path assay has an interesting pattern called SGTF when on that B117 variant. So the TAC-path assay has 3 different probes sets that recognize N, ORF1AB, and spike gene sequences. B117 has this deletion, and so you don't detect the S probe. So you'll get positive detection on these other 2, but you'll have what's called S gene target failure, or SGTF. And this has been a useful surrogate for B117 in tracking its frequency in regions of the US, as is shown here on this graphic from CDC, looking at SGTF and non-SGTF samples over time. The important thing to realize is that there are other variants that also have this deletion. And so there's been non-B117 in the US that also will give you SGTF. And it's just important to realize that this is a surrogate marker. So it's not always diagnostic of a given variant because these mutations can occur in multiple variants. This is a useful website. If you just Google FDA guidance and viral mutations and tests, you can find it. It has up-to-date information on molecular tests and how they're impacted by SARS CoV-2 mutations. And then, of course, now there's more commercially available primer probe sets to detect specific mutations. And as I indicated, of course, mutations can be found in multiple variants. So here's a primer probe set that would recognize N501Y and on this graphic, and you can see that it's found in various lineages at varying frequencies. So it's not entirely specific for a given variant, but is useful as a marker. Similarly, E484K is in many different lineages now, and many different variants. And so it's a good marker, and it's a good probe to use, but it's not entirely specific. And so, for example, if you want to detect B117, people will actually use this ND3L mutation because it's only really present in B117, so it's a reliable marker for that lineage. And then, of course, quality control in these PCR assays is also important because you're often dealing in surveillance with something that might be rare at first. And so if something is rare or low prevalence, your assay has to be extremely sensitive and specific to have a good predictive value, something I'm sure many of you are familiar with in diagnostics. And this just, again, is a reminder that if you have some variant that might be at 10 or 20% prevalence, even with a 90% sensitive and specific assay, you're still basically at 50 to 70% predictive value. And so it just really highlights that even minor decrements in sensitivity and specificity can really diminish the utility of an assay that's not well quality controlled. So key points in evaluating diagnostics are to determine the analytic sensitivity and specificity. In qPCR assays, this would be how well you can discriminate between alternate alleles. So how well do you discriminate 501Y from 501N at the N501 mutation? How do you determine the cutoff CT or your limit of detection? And then in all these things, synthetic materials like these RNA templates are often more readily available than clinical specimens, particularly for newly arising variants, and they are more easily adapted to a quantitative readout, and that you know exactly how many copies there are in your template for quality control in these assays. And then also high accuracy is necessary when mutations or variants are rare. And then matching mutations to the actual variant is important in evaluating the diagnostic utility. So I'll spend the next few minutes talking about the clinical impact of these variants. One is looking at monoclonal antibodies. So there's three different monoclonals currently that have FDA emergency use authorization for treatment of individuals infected with SARS CoV-2. This is quite a complicated process because they are only really effective when they're given very early in infection. And so there's, again, three. One is casirivimab plus imdevimab. That's called the Regeneron cocktail. There's bamlanivimab, or BAM, which is an Eli Lilly monoclonal. And then Eli Lilly now also has a cocktail, bamlanivimab and etazivimab together. And the good thing about these cocktails is they have two monoclonals that recognize different parts of the spike protein, and so they are more likely to retain neutralization activity even in the presence of a specific mutation. And these tables are from the EUA fact sheets from the FDA. You can find it on their website. And it shows for the different variants, the key substitutions in them, and what that does, how well the antibodies recognize them. So you can see that the casirivimab and imdevimab cocktail works well against many of the most common variants, whereas E484K or L452R really make it so that bamlanivimab doesn't work very well against those viruses. And so a patient who has one of these variants who's treated with this antibody may have less of a response or get less benefit. So, however, if you have the cocktail of BAM plus etazivimab, you retain activity against many of these variants. So at our center, my laboratory has been doing a lot of sequencing, and we provide weekly reports on the frequency of given mutations and variants in our patient population. And this is just one of them, showing the frequency of N501Y here in green, which is a marker or a mutation on B117, and this is when B117 really surged in Michigan. But then lower down, you can see the red line, which shows L452R and its frequency over time, and E484K in yellow. And what we do is we work with our antimicrobial stewardship team These are just two tweets from Jason Pogue from our group. And we track these mutations, and once we're at a specific frequency, we'll alter what we use as our empiric therapy for monoclonal. So if we see a lot of L452R and E44K, we might switch to either the Eli Lilly or the Regeneron combinations as our empiric therapy. So this is just one way of using surveillance data on viruses in a community to help choose empiric therapy. Even if you can't, on an individual level, get the identification of the variant quick enough to affect antibody choice, you can do so at a more population level. So what about vaccines? And so this is a headline from a New Yorker article early in January when there was a lot of buzz about variants in vaccines and concern that the variants may evade the vaccine. And since then, there's been a torrent of data on this that I'll attempt to summarize in just a few minutes. So again, spike is the major antigen that we're talking about here. It's the glycoprotein that sits on the surface of the virus. It mediates receptor binding and entry into cells, shown in the red picture. But it's also recognized by the antibodies, in that if an antibody binds the spike protein, it'll neutralize the virus, and then the virus can't bind receptor, so they would be protective. There's been a lot of data, a lot of studies looking at how well someone's antibodies recognize different variants. So of course, the vaccines that are in use uniformly use spike protein from what might be considered the original or ancestral SARS-CoV-2 that was around in spring 2020. Now the spike variants are different, and so people have taken sera from people whose immune systems have been stimulated by this original variant, in this case WA1, Washington 1, shown in gray, and then compare how well those sera recognize other variants. And here showing B117, P1, and B1.351. This is just from a couple studies that are representative and some of the first, and looking at how well these sera from vaccinated individuals recognize spikes. And you can see that B117 appears to be well recognized by post-vaccination sera, even though there's a dramatically different spike protein. So it's not terribly different antigenically from WA1. However, P1 and B1.351 are a little bit different, and P1 appears to be nearly as well recognized as the other variant, B117 and the original. But B1.351, there does appear to be a drop-off. It's about 3 to 4-fold in most studies. And so there's a drop-off, but what we don't know is, does that translate to not being protected? Then here's a similar data from another study looking at B1.351, B1.429, which is one of the, quote, California variants, and then looking at a range of vaccines. So this would be convalescent sera from people who recovered, the Moderna sera from people who got the Moderna vaccine, or sera from people who got the Novavax subunit vaccine. And you can see that with 1.429, there's a slight decrease, similar to P1 perhaps, and then B1.351 has a several-fold, approximately 5-fold decrease in neutralizing activity. So it appears that B1.351 in particularly is antigenically different, that if you stimulate your immune system by vaccination with the original SARS-CoV-2, those antibodies aren't quite as good at recognizing B1.351. The big question is, how does this affect protection? Because we don't know what titer is really protective. This is data from an animal study with monkeys looking to try to define that threshold. And so you have an antibody titer of approximately 40 is protective of macaques from infection. That means you can dilute someone's sera 40-fold and still show that that sera would still be protective. And so that helps place some of this in context. I'll just go back to an earlier slide. So we see that with B1.351, you get a drop in your sensitivity shown in this plot here to neutralization. And the question is, are you still protected? Are you still above this threshold, which is at this dashed line of 1 to 40? And so that's reassuring that many people will still be protected, even if they are presented with B1.351. But this is an area of active investigation, and we still don't really know for sure. So moving ahead, it just highlights that looking at sera can give you some sort of idea at how well a vaccine may work, but it's not everything. And shown here are a set of titers, which are actually my antibody titers against different flu strains. And so you can see that I have quite good titers against the vaccines, which are the egg-based flu, at 1 to 2560. You can dilute my sera 25 to over 2,000-fold, and it'll still neutralize these viruses. However, it's not quite as good against these actually circulating viruses, which are labeled as cell. Now, does that mean I'm not protected? Well, I probably do have some level of protection because I actually haven't gotten symptomatic with flu in the past 10 years. And that just really highlights that the serology is only part of the story. And it's also that vaccines don't really work as a plus minus, that it's also, does it protect you from severe disease, mild disease? Does it prevent transmission? What are the population effects? And it really gets complicated. So what do the actual studies show? Because the best way to show whether whether a vaccine doesn't work against the variant is to see if the vaccine doesn't work against the variant. And the data now are accumulating. Here's a slide that I try to routinely update as papers come in. I'm looking at some of the data from the randomized trials and some of the observational studies as well. So the Janssen or J&J vaccine in the FDA filing, which is now published as a peer-reviewed article, it was 72% effective in the United States. It retained a fair bit of that efficacy in Brazil, where the P1 and P2 variants were circulating. It was 68% effective. And there was also an arm of the trial, excuse me, in South Africa, where it proved to be 64% effective, and that many of the cases in the vaccinated arm of the study were due to B1.351. The Pfizer mRNA vaccine, that trial had an arm that was performed in the United Kingdom at the time that B117 was becoming prevalent. And there's also been the Siren study, which is an observational study in the United Kingdom at that time. The Siren study showed that it was 85% effective in protecting against infection, which was presumably when B117 was prevalent in the United Kingdom. And then Novavax had a similar, its study was going on in the United Kingdom when B117 was around, and it was quite effective. And then it had some drop-off in efficacy against B1.351 from the study arm in South Africa. And so there's almost every week there's new data on how well the vaccines work against these variants. It seems that B117, there's going to be retained vaccine effectiveness, but that there might be a small drop-off in protection from B1.351, potentially P1, but these vaccines still prevent severe disease from these variants as well. And that kind of charts the way forward. Much like in influenza, there will be surveillance every year to look at how different variants that might be around affect how the vaccines work. And so you can do this by looking at people who are vaccinated and still get infected and try to see if the variants are more common in those individuals, which might suggest that they are evading the vaccine protection. And this is just one case report in the New England Journal looking at a breakthrough infection. The challenge here is sometimes these numbers are small. So this report looked at 2 people who had breakthrough infections, But to really understand how these viral sequences relate to breakthrough infections in vaccinated individuals, you really need large numbers. And so those studies are ongoing, and I suspect within the next year we'll have a lot more information on how variants are affecting vaccines. And so I'll conclude by thanking you for your attention, and look forward to discussing this further in the Q&A. Thank you.
So let's begin. Thank you, Dr. Lauring, for that informative presentation. We will now start the Q&A portion of the webinar. And if you have any questions you want to ask audience members, please do so now. Just click on that Ask a Question box located on the far left of your screen. So let's get started. It looks like we already have some great questions coming in from our audience. Dr. Lauring, how closely should labs be monitoring the proliferation of variants in their populations?
Sure. I think it's going to be important moving forward that clinical microlaboratories, keep updated on, not only what's going on nationally, but what's going on regionally in their catchment areas, because as we've seen, there's going to be new variants coming, and it's important to be aware of it, and of course, be ahead of the game in terms of any measures that need to be doing to either update the assays or know how your various testing platforms are going to perform, in the setting of a new variant. So I think it will continue to be important, at least for the foreseeable future.
Thank you so much. And that brings us to this next great question from one of our audience members. What action should be taken to help challenge our assays to make sure they are detecting existing and any new mutations? Sure.
I think, one thing is, as I indicated in my talk, the FDA is continually updating on their website as data come in on how various approved or EUA diagnostics are affected by variants. But I think labs will certainly have a need to test their assays against new variants as they arise, either by having actual available residual clinical samples that correspond to given variants, or by perhaps quality controlling their assays on defined templates or RNAs that have the same sequence as these variants, just to ensure that the assays are still performing as expected in terms of diagnosing SARS-CoV-2 in someone who may have a different variant.
Thank you so much. And again, I want to thank our audience for these great questions coming in. Our next question, aside from the public health value, is there a clinical role for distinguishing SGTF from non-SGTF variants?
Sure. I think the main clinical role would be in terms of therapeutics. Even the vaccines, I would consider that kind of half clinical, half public health, because often you would be dealing with someone who's vaccinated, and it's of interest to know if they have a variant, and that's why the vaccine may not have worked. But in terms of direct clinical utility, I think it's therapeutics, and the area where it's become most important, as I indicated in the talk, is with antibody therapy, where that can make a difference. And so I think that's going to be a key area. We don't really have a lot of information on variants and other therapeutics, but that certainly is something that could play out in the future. Whether certain variants, as we've seen with other viruses, are less sensitive to remdesivir or other antivirals that come down the pike, that will become important as well. And then as a clinician, I can tell you that even if it's not clinically important, your clinicians will be asking you, asking the clinical lab, is this, variant X? Is that why this patient, is not doing well? Even if it's not really usable in that way, you'll find your clinicians are going to want to know. And then the last thing is in the clinical realm, but also public health, is we found that knowing variant status is of some use to our infection control and prevention teams in our health system in terms of making sure that they're doing everything they can to limit the spread of SARS-CoV-2 and COVID in healthcare settings.
Thank you so much. Now, do you see the need for developing variant assays and submit for EUA?
I think there certainly is a role for it. Obviously, I'm not a regulatory specialist, but I think you can certainly see, particularly if we find more and more that these variants can affect, clinical decision making, that having an EUA, for detection of a specific variant would be important, so that has a layer of compliance and regulatory, so that you can use that data and make decisions based on it. So I do think there's a role. How expansive that role is, it's going to be. It's hard for me to say right now, but I do think it's important.
Thank you so much. Now, what role will PCR assays detecting mutations will have, particularly in complement to sequencing?
Sure. I mean, I think you can see it playing out kind of two different ways, and I do think phrasing it as complementary is really key. Of course, sequencing is unbiased, right? It's going to detect what's out there. Whereas PCR is a much more scalable and directed way for specific variants. So I think both are going to be hand in hand. I think once a variant is in an area at a certain frequency, I think PCR is going to be the way to go. And we've certainly seen this with B.1.1.7. Which was first identified, and tracked from sequencing, and then it was realized that it had this unique signature with the TAC path assay, as I highlighted in the talk, and so then that became a nice surrogate, to really know very quickly who would have that variant and how it might be use that information moving forward. So I think they work hand in hand.
Thank you so much. And we have a final question from our audience members. They say, great talk, covered in detail about the impact of the mutations on the vaccines. How are the mutations impacting the diagnostics? Any thoughts on how best we can quantify that impact?
So if I'm understanding, the question is, how can we measure how much, either one variant or any number of variants are affecting our ability to diagnose SARS-CoV-2? I must say I haven't seen much evidence that you're getting, in a significant manner, like complete assay failure, that your assays are not detecting. And I've seen a few reports here and there, but in terms of the prominent variants, most, if not all, of the diagnostic platforms will still give you a diagnosis that, even the TAC path assay, which has this signature of the S gene target failure. There's two other targets, and those are picking it up well. So I'm not aware of any of the variants yet having a significant impact on just overall diagnostics.
Dr. Lauring, thank you for your presentation today. Would you like to provide any closing remarks to our audience before we go?
No, I think it's an exciting area, and it's something to continually watch because it's really an area where I think clinical diagnostics are going to be potentially rapidly changing over the next year or two.
Absolutely, and thank you again for your time today and for your important research. Before we go, I want to thank our audience members for joining us today and for their important questions. Questions we did not have time for today will be answered and posted in the event resources by June 4th. And this webcast will be available for on-demand in the auditorium from Thursday on. If you found this information informative and helpful, please share it with your colleagues. And for the next webinar on June 30th, we will have Dr. Lauren Bachman from Virginia Commonwealth University Medical Center presenting on best practices to recover from an out-of-control event. Please stay tuned and watch your email inbox for that invitation. Until next time, everyone, stay safe and stay healthy. Take care. Bye-bye.